Okay. Now we will see the next chapter, that is biotechnology and its applications. Okay. How are biotechnologies useful to us? In what field? Yes, okay. Yeah, in therapeutics, yeah, in diagnosis, and then genetically modified crops for agriculture and processed food also. Okay. Now let us see how is biotechnology application in agriculture. Okay. To feed the increasing population in the world, we should also increase the food productions, and there are some methods to increase that. The first one is agrochemical agriculture, and then organ agriculture, and genetically engineered crops based on agricultures. Okay. Now let us see about genetically modified organisms. What are genetically modified organisms? We saw in last chapter, right? Yeah, that is correct. What they do is then organisms which result with the gene is taken from one organism to the other organisms in the laboratory for improving its function, ability, and for obtaining a desirable results whatever we want. And then these genetically modified organisms are called as transgenic animals. Okay. Okay, we can include any type of organism like any plant, bacteria, or even microbes, and whose genes have been altered according to our desirable effect. Okay. These type of genetic modification had made so many changes in the agriculture field, like what crops became more tolerant to abiotic stresses like extreme temperature or extreme cold or extreme heat, and then dependence on the chemical pesticides has been reduced. Okay. And that is pest resistant crops. It helped to reduce the pest harvest losses. And then it also increased the efficiency of minerals, yeah, and whatever minerals and in plants, and preventing what? Early exhaustion of fertility of the soil. And it also enriched it also enhanced the energy rich products, energy rich vegetables, fruits, anything. Okay, now let us see about biopesticides. It is an application of the agriculture. Biopesticides is one of the application in biotechnology agriculture. What is that? Yeah, it is a production of the pest resistance that decrease the yeah, that's correct. Bt toxin is produced by a bacterium called Bacillus thuringiensis, and example Bt cotton, Bt corn, rice, potato, tomato, and example soya. Yeah. Okay, now let us see about transgenic cotton, that is Bt cotton plants. With increased sprayings, these caterpillars, what they do is build up their community due to natural selection. After this Bt cotton, what they did, the genetically modified and contain a gene code that production of the codes for the production of the protein that kills the caterpillar, okay, that kills the caterpillar of the Helicoprasa mouth. So, for that killing this type of caterpillars, this Bt cotton was made. And the next is pest resistant. Pest resistant is, what is that? It is a production of large crystal like protein structures which have a insecticidal activity. And example Bt toxin, what is, the crop is infected by some European corn borer. What they do is the gene is inserted into the cone, the crop. The what gene? Bacillus thuringiensis, and pest dies when feeding on the any part of the plant. So, after the insertion of the gene, whatever insects or whatever pest which feed on them automatically die. Yeah, because of yes. Okay. Now let us see how biotechnology is useful in medicines. The recombinant DNA technology process have made an immense impact in the area of healthcare, enabling a mass production of mass production of safe and very effective therapeutic drugs. Yeah, the recombinant therapeutics do not unwanted immunological responses as in common cases of similar products which is isolated from non-human sources. At present, about 30 recombinant therapeutics have been approved for human use all over the world, and out of these 12 are from India. Okay. Now let us see what is a genetically engineered insulin. Okay, in older days, where we get insulin from? Like which animal? Huh? Insulin from which animal? From the pancreas of a pig. Okay, pig. Okay, this insulin consists of two short polypeptides. One is A chain and another is B chain. Okay, which is linked together by a disulfide bridges. How many chains?
2 chains. A chain.
B chain, which is linked together by a bridge called disulfide bridge. Okay. In mammals, including humans, these insulin is synthesized as a prohormone only. Okay, synthesized as a yes, which contains a extra stretch called C-peptide. Actually, it's not given there. Okay, the C-peptide is not present in mature insulin and removed during the maturation into insulin. So, what happens? Therefore, a main challenge for the production of insulin using DNA technologies was getting assembled in this technology. So, in 1983, that is Eli Lilly, an American company, prepared the 2 DNA sequences corresponding the A and B chain of the human insulin, and which introduced them in the plasmids of E. coli to produce the insulin chains, and these chain A and B were separately extracted and combined by creating a disulfide bond to form humans, and that is called as humulin. Okay, and see, this is a pro-insulin in A peptide, that is A chain and the B chain. It converts into insulin and the bridge in the bridge of C peptide. Okay. Okay. What is a recombinant vaccine? What is recombinant vaccine? Vaccines produced through genetic engineering are called as what? Recombinant vaccines. The vaccines produced through genetic engineering called as recombinant vaccine. Okay. Like example vandu hepatitis B vaccine and then polio vaccine are the examples for recombinant DNA vaccines. Now let us see the example for disease. For viral disease, recombinant vaccines have been produced are chicken pox and then genital ulcers, hemorrhagic fever, acute infantile gastroenteritis, and in bacterial diseases it is cholera, pneumonia, tetanus, tuberculosis, typhoid, meningitis. Parasitically it is malaria, filariasis, sleeping sickness. Idukella po enna vaccine ivangalla po? Yes. Okay. We will see about stem cells technologies. What is stem cell technology?
Stem cells எடுத்து இன்னொரு டிசீஸ் வந்து ட்ரீட் பண்றது.
Okay, அப்ப stem cells na என்னது?
அது வந்து தொப்புள் கொடியில இருந்து
எடுப்பாங்க. Correct. They are the undifferentiated biological cells. நீ சொன்ன மாதிரி biological cells that can differentiate the specialized cells and can divide to produce many stem cells, and stem cells are of two types. One is embryonic stem cells and another is adult stem cells. Okay, embryonic and
adult.
Yes. Embryonic na isolated in the inner layer of the blastocytes, that is embryonic, whereas in adult na which exits throughout the body from the embryonic development. அது வந்து embryonic stageல மட்டும் இருக்கும், மாத்திரம் இது வந்து throughout the body, throughout the body after the embryonic development, and found inside different type of tissues like example brain. Okay, applications of stem cell cells. எதுக்கெல்லாம் யூஸ் ஆகுது stem cell cells? Like specific என்ன type of disease? organ and tissue regeneration, and then bone marrow transplantation, and also like cardiovascular disease or a treatment ku adukaprom blood disease treatment also, and cell deficiency therapy. Bone marrow transplantation, and then cardiovascular disease treatment, அப்புறம் blood disease treatment, அப்புறம் organ tissue regeneration, அப்புறம் cell deficiency therapy. Okay. Okay. Now let us see what is a gene therapy. It is a collection of method which allows the correction of the genetic defect, and that is being diagnosed on a child or a embryo. ஒரு childல ஒரு genetic gene வந்து defective இருந்தா அதை கண்டுபிடிச்சி what they do, alter or extend whatever they do, it's called as gene therapy. Like example, இப்போ வந்து cells with a non functioning gene, ஒரு cell வந்து gene வந்து function ஆகல, and what they do, adding the DNA containing a functional version, அப்ப அந்த geneல எது இல்லையோ அதை வச்சு insert they are adding that, and which the cells function normally. This is called as gene therapy, and the first gene therapy was done in 1990. Okay, in a 4-year-old child with the adenosine deaminase deficiency. Okay, and let us see the example for gene therapy in cystic fibrosis, in vivo and ex vivo. In vivo na it is inside, exona outside. Okay, first enna பண்றாங்க, ஒரு இது வந்து டிஃபெக்டிவா இருக்காங்க, insert the transgene or a into a liposomes or a lipophase or a viral இது அது specific gene வந்து insert பண்ணிட்டு deliver to the site of the action, that is in lung by a aerosol spray. இது வந்து என்னது inside, and the outside enna பண்றாங்கன்னா they remove the specific cell which is defective actually, and enna பண்ணுவாங்க grow cells ex vivo, and then add the transgene and select the modified cells and replace the modified cells with the defective one. Okay, now let us see molecular diagnostic techniques. For effective treatment of the disease, the first enna key point, first the diagnosis is ரொம்ப important to treat a what specific diseases. So now let us see the methods, some methods for diagnosis. Okay, the first one is PCR, that is polymerase. Yeah. PCR enna panna, this helps in the early diagnosis or early detection of a specific disease, and this technique can be used routinely to detect HIV. Okay, and in suspected AIDS patient. It is also used to detect mutation in genes that is suspected in cancer patients, and also a powerful technique to identify several genetic disorders. இது என்னதுக்குல யூஸ் ஆகுது?
HIV detect பண்ண
Yes,
AIDS patient
அது AIDS patient ரெண்டு ஒன்று. Okay, mutation in அப்புறம் various genetic disorders. Okay, and the next technique is recombinant DNA technology. இது என்னன்னா, it is a molecular diagnostic feature done in following steps. என்னென்ன ஸ்டெப்ஸ்னா, first வந்து a single stranded, that is DNA or RNA, is tagged with a radioactive molecule called a probe, which is allowed to hybridize to its complementary DNA in clone of the cells. அப்புறம் the defective cells are detected by a autoradiography. அப்புறம் last step என்னன்னா, the clone is mutated with, and the clone with mutated gene is not observed on a photographic film because the probe doesn't show any complementary with the mutated gene. And the next one is enzyme linked immunoassay. இது எங்க யூஸ் பண்ணுவாங்க? இது லைக் எந்த டயக்னோசிஸ்ல? எலிசா. என்ன டிசீஸ்? எய்ட்ஸ். Elisa, yeah, AIDS. Okay. This is a diagnostic. This is also another type of diagnostic disease. I mean, what technique in which the principle is based upon a antigen antibody interaction. Infection by the pathogen can be detected by the presence of antigens like proteins and glycoproteins, or by detecting it antibody synthesis against the pathogen. Okay, the next is transgenic animals. What is transgenic animals? DNA? Yes. Animals that have DNA manipulated to process whatever the desired we want. Okay. These are known as transgenic characters. Yeah. Like example, transgenic rats, and then rabbits, pigs, sheep, etc. Over 95% of transgenic animals are mice only. Okay, now let us see the benefits of transgenic animals. There are several, and the first one is normal physiology, then development. What in transgenic animals, they have a specifically designed to allow to study the how genes are regulated in them and how they affect. It's for what? Like studies, experiments, they are very useful. And another, I mean, the study of the disease. And then next is biological products. Produce useful biological products can be treated to the introduction of the portion of the DNA, and then such as human protein, which is to treat emphysema. And the idilende number we can treat various disease like emphysema. And then vaccine safety, yes, like example polio vaccine. And then chemical safety, na, it is also known as the toxicity or a safety testing. It is similar procedure used for testing toxicity of the drugs. If ஒரு drugs ஓட toxicity கண்டுபிடிக்கணும்னா we can rather use these kind of transgenic animals. Okay, and the next one is patents and ethical issues. What is patent? I mean patent. Yes, ama. It is a set of exclusive rights granted by the government to the inventors or an assignee to prevent others from the commercial use of their invention. That is patent. Okay, then what is ethical issues? Okay, Indian government has set up for an organization called Genetic Engineering Approval Committee, which makes all the decision regarding the validity of the research and the safety of introducing the genetically modified organisms for public services. That is ethical issues. Now let us see what is about biopiracy. What is biopiracy means? It refers to the resources of multinational companies or the other organisms without a proper authentication from the countries, authorization from the countries and the people concerned with the compensatory payment. Now let us see what is. It is a practice commercially exploiting by natural occurring biochemical or a genetic material, especially by obtaining patent that restrict its future use, and while failing to pay their compensation for the community from which it originates. Okay, அந்த பர்டிகுலர் கண்ட்ரியோட அது ஒரிஜின் ஆயிருக்கும், பட் அவங்க கிட்ட இருந்து பெர்மிஷனோ பேட்டன்ட்டோ அந்த காம்பன்சேஷனோ கொடுக்காம இவங்க எடுத்துக்கிறது மல்டிநேஷ்னல் கம்பெனிஸ். ஓகே, அண்ட் பயோ ப்ரோஸ்பெக்டிங்னா the search for biological resources and accompanying indigenous knowledge for the purpose of commercial exploitation. It is a process of appropriation and commercialization of natural products ranging from plants and animals. It is legal but may promote, bio prospecting is legal but many may promote biopiracy. Okay. Now let us see the see about a controversy that happened in India regarding a biopiracy. Okay. And basmati rice, இது வந்து ரொம்ப ஒரு பெரிய பேசப்பட்ட கான்ட்ரோவர்சியா. என்ன ஆயிடுனா, அமெரிக்கன் கம்பெனி லைக் கால்ட் as ரைஸ் டெக் 1, அது ஒரு கம்பெனி. ஓகே, என்ன பண்ணாங்கன்னா, they won a patent on the basmati rice, and the basmati rice which was originated from India only. So இது வந்து ஒரு பெரிய கான்ட்ரோவர்சியா ஆச்சு. ஓகே, and the next one is ஒரு நிமிஷம். Yes, turmeric, correct. In May 1995, the US Patent Office granted to the University of Mississippi the patent of use of turmeric in wound healing. But the turmeric one, originally where it originated from? India. Yeah. And the next one is what? Do you know anything? யா ஃபங்கிசைடல் யூஸ் ஆஃப் நீம் ஆயில். அதே மாதிரி வேறு கம்பெனி ஒரு பேட்டன்ட் வாங்கிட்டாங்க, அதோட இது வந்து இட் ஒரிஜினேட்டட் ஃப்ரம் ஹியர் ஒன்லி. ஸோ தீஸ் ஆர் த கான்ட்ரவர்சிஸ்.